Hello, wonderful AP Bio students, and welcome to our video on DNA replication. Now, this is exactly how it sounds. We take one DNA and we replicate it and we get two DNA. And when does the cell need to do this? Well, the cell needs to do this when we take one cell and we go through our favorite process called mitosis, and we end up with two identical cells. So those two identical cells need to also have identical DNA within them. And actually, it's kind of crazy, but 6 feet of DNA is coiled up in each of these little microscopic nucleus of cells. So that DNA needs to be duplicated in each and every cell that goes through mitosis within our body. And cells need to go through mitosis so that we can grow and we can repair and we can have new cells when others die. So let's take a quick look at DNA first, because it's going to be important to know all of the parts. So this should be a review. DNA has two main parts. It's got this like backbone part right here. This is what we call the DNA backbone, and there's two of them. And the DNA backbone is made up of a phosphate group, and it's also made up of a deoxyribose, this pentagon here. And a deoxyribose is a 5-carbon sugar. And so a lot of times you'll hear, oh, your DNA has sugar in it. Well, it's not like table sugar. However, deoxyribose is it is a sugar. So we have a 5-carbon sugar. And these two are held together by pretty strong phosphodiester bonds right here. And that's what holds the phosphate groups and the ribose groups together. Then what we have are our nitrogenous bases. And we have two types of nitrogenous bases. We have purines and we have pyrimidines. And purines are 2-carbon chain nitrogenous bases. They're called nitrogenous bases because, yes, they are carbon chains, but they also have a lot of nitrogen in them. And then a pyrimidine is just a 1-carbon chain nitrogenous base. And these, a purine always pairs with a pyrimidine. In this case, the purine guanine is pairing with the pyrimidine cytosine. So guanine and cytosine always pair together, and adenine and thymine always pair together as well. And the way that they pair is through a hydrogen bond. Now, hydrogen bonds in DNA are relatively weak, and we are going to be looking at what it takes to break these bonds. So we can see that between the adenine and the thymine, we have 2 hydrogen bonds, and between guanine and cytosine, we have 3 hydrogen bonds. Another thing that's pretty interesting about DNA that's important to understand is the fact that DNA is antiparallel. Now, if we were to say, okay, DNA is parallel, then we would be looking at these nucleotides right here, and these nucleotides have a specific shape. And a lot of times we represent this shape as an L because it has 3 distinct parts. So those would be the points of the L. And those 3 distinct parts are the phosphate group, the ribose, and the nitrogenous base. When we look at a 3-part nucleotide, which is the monomer of DNA, so I'm going to write this is a nucleotide, which is the monomer of DNA, we could see that this is repeated over and over and over again. It's basically phosphate group, ribose, and one of the 4 nitrogenous bases. And if we were to say that DNA was parallel and not antiparallel, we would assume that the nucleotides, maybe line up like this, would also line up the same way on the other side. But this is not the case. Where's my red? Because there it is. A big no sign. This is not the case because the way that the DNA is set up and the way that all of these components bond together, it arranges itself in kind of a mirrored direction. That's why it's called anti-parallel. So if this L is facing like that, the other L is going to be kind of mirrored upside down. So we look at the nucleotide of DNA, they actually run anti-parallel together. So we could do this with our fingers. We have, L for love, and instead of connecting together like this, we have one side of the DNA that is oriented in a different direction. And those continue to stack on top of each other and create our row of DNA. So another thing that's pretty interesting, because we have our antiparallelism, if that's a word, let me clear this off here. We also have, let me get blue here, we have a direction of DNA. In other words, direction has, or DNA has a clear end and beginning. And the beginning is a 5'. Now we call this 5' because 5 phosphate. It really has to do with the carbon number that the phosphate group is headed off of. But we call this 5' end, and that's typically the starting end. And DNA ends with a 3'. And the 3' end always ends with this hydroxide group. So we have 5' phosphate, 3' hydroxide. And because DNA runs anti-parallel, the other side is going to be the exact opposite. So we have a hydroxide group right here, and so this is going to be our 3' end, and this is going to be our 5' end. So if we were to say that DNA has a direction, and that direction is always 5' to 3', well, the direction of this side of the DNA would be that way, and the direction of that side of the DNA would be that way. And this happens to be the direction of replication as well. So let's jump right into replication. Replication, in DNA replication, we use what's called the semi-conservative replication model. And that's a super weird, almost political sounding name. But when scientists began to look at this replication, they were trying to figure out the way in which the DNA was replicated. Was DNA simply the whole thing put into a copier and then you have an original and you have a copy? Or do you have a copy that has parts of the original in it? Since DNA is coded, we can always make correct new pieces to code for it. So what is the best theory of how DNA is made is a semi-conservative replication model. And how that starts is we have, and I'm going to very much oversimplify this, but this is just to give us an idea. So we start with our DNA strand, and this is going to be our, what we call our parent strand. Parent strand. And this is just a very small section of DNA, and these are our nitrogenous bases. We have two making up our, here would be our hydrogen bonds right here. And our nitrogenous bases, let's just call them thymine, adenine, guanine, cytosine, and on. Since thymine always pairs with adenine, we'd have an adenine over here, thymine over here, guanine always pairs with cytosine, and cytosine always pairs with guanine. Okay, so this is our parent strand. Now when we go under replication, the parent strand becomes unzipped by this enzyme called DNA polymerase. And DNA polymerase unzips 1, 2, 3, 4. Let's see, we only had 4 there. 1, 2, the DNA. So I always say DNA polymerase is kind of naughty because it likes to unzip things. So here is DNA polymerase. And that is the enzyme. Remember, it ends in A's. That's the enzyme that actually breaks apart those hydrogen bonds between the nitrogenous bases. So now we have kind of 2 half strands of our parent DNA, and we still have our TAGC and our ATCG over here. So now there's these exposed bases. And these exposed bases want to pair up with another nucleotide because they're willing to make a hydrogen bond very easily. And we'll learn why they don't actually just bond right back together a little later. So this is our DNA is split, and then we have the cell has manufactured. So here's our 2 strands right here. 1, 2, 3, 4. The cell has manufactured and has kind of floating around these free floating nucleotides. Now remember, we use little L's to symbolize nucleotides. So if this is our phosphate and this is our ribose, let's say our nitrogenous base would be adenine here, thymine here, guanine here, cytosine here. And these free-floating nucleotides, through the use of enzymes, etc., which we're going to learn about, will eventually pair up with 1/2 of the parent strand. And I'm making these antiparallel. So in semi-conservative replication, you take the parent strand, and a piece of the parent strand is found in each of these copies. It's almost used as a template so that the free-floating nucleotides can use it to code the other half. So these strands right here, these two strands, they're identical, and they're called identical daughter strands. And these identical daughter strands have half of it is the parent strand, and the other half is essentially new material, although it's coded for the same, if that makes sense. So instead of taking a, here's my really quick DNA, instead of taking DNA and having, as your result, you still have two parent strands, but one is the parent and the other is almost like a photocopy of it, we see that DNA actually is partially the parent and partially the copy. So that's what semi-conservative replication means. So now we're going to look at the nitty-gritty. You have a piece of paper that looks like this, and I'm going to be writing things on it and then erasing it, and then writing things on it, erasing it, just so that I can make it big enough for you. And we're going to look at kind of the mechanisms used in DNA replication. So this is the paper that you should be taking your notes on. It's also linked on this page, and I did pass it out in class. So make sure that you have this handy because you're going to need this picture. So this is we're zooming in on a special little strand of DNA. And I want to do a few labeling things first. So we're going to label this part right here 5'. So that means that at the very end here, what's this going to be labeled, the dark blue at the end? You're right, 3' at this end. So what would that make this end? That would make it 3'. And we follow that side of this strand all the way to the end, and that would make it 5'. So we can look at this, and so this would symbolize the new material, and this would be 5'. And this would be 3'. Okay, so now that we have that out of the way, our first step is going to be looking at this area right here. This area is called the replication fork. And this is where replication is happening. So what causes the replication fork is this special little enzyme that actually unwinds the DNA, and that is called DNA helicase. So DNA helicase unwinds DNA, and those are proteins which were coded for by the DNA. So now that this is what's actually separating those strands of DNA, how come these hydrogen bonds, which if these little teeth, they kind of look like teeth, if these little teeth represent the nitrogenous bases, how come they're not just ready to pair up with each other again? Well, we have these specialized proteins. You could see them on here, and they actually go along the whole strand, but we only have a few pictured here. These are called binding proteins. So in our step 2, we see these single strand binding proteins is what they're called. Single strand, and I'm going to write into number 3, but you should have room to write this within your number 2 space. Binding proteins, and what they do is they stabilize the unwound DNA. And they make it so that these hydrogen bonds don't rebond together, and they keep that replication fork open. Okay, so now I think I've filled up enough. Hopefully you've got this. If you don't, go ahead and press pause right now because I'm going to erase this. So now what we have, let's look at our step number 3. We have this special awesome protein right here, and this special awesome protein is called DNA polymerase. And DNA polymerase is kind of like the machine. I call it kind of like a, what is it called when an assembly line. So the DNA polymerase makes sure that the assembly line is happening, and it attaches these free-floating nucleotides to their specific pairing nucleotide. And this is what we call the leading strand, because we are moving in a 5' to 3' direction. In fact, the direction of replication is going to be this way. So direction of replication. So it's actually going towards the fork is how replication works. So we are moving in a 5' to 3' fashion, and it's really easy on what we call this strand right here, which does go from 5' to 3'. This is called the leading strand, and you'll understand why after we talk about the next strand, which is called the lagging strand. So the leading strand is synthesized continually, going from 5' to 3' by, who is it synthesized by? Well, it's synthesized by DNA polymerase. So DNA polymerase is a superstar enzyme that's gathering all these free-floating nucleotides, pairing them up, and actually making that bond happen, making those hydrogen bonds happen, and those bonds between the deoxyribose and the phosphate groups happen as well. So it is effectively creating one of our daughter strands. Well, that is the leading strand. Well, we run into a problem with this guy right here. This guy is called the lagging strand, and he's kind of like the redheaded stepchild of DNA, because the lagging strand, it's got issues. All right, I'm not saying that every redheaded stepchild has issues, but the lagging strand, it's a little more difficult to create the daughter self or the daughter DNA from the lagging strand. So hopefully you got all this. If you don't, pause it. I'm going to erase it right now. So our lagging strand actually is replicated not continuously, but discontinuously. So if this is our poor little lagging strand right here, lagging strand is synthesized discontinuously. I'm going to write this all out, and then I will explain it using the diagram. So it's synthesized discontinuously, and what happens is it does it in short little sections, and it uses this thing called an RNA primer, and it makes these Okazaki, these little special little fragments called Okazaki fragments. So it's synthesized discontinuously. Primase synthesizes a short RNA primer, which is extended by DNA polymerase to make an Okazaki fragment. And this, of course, is named after the guy who discovered it. So, what does all this mean? So the lagging strand is synthesized discontinuously. It's synthesized in little choppy fragments, which is then put together again by DNA ligase. So let's look and name our parts. So this right here is this red section right here, and actually I'll change my color to red. This is an RNA primer. So this is actually made of RNA, and it attaches itself to some exposed base pairs to kind of give a foothold, if you will, for the rest of the DNA nucleotides. So we have our RNA primer here. Since DNA can only be replicated from 5' to 3', we're looking at this section right here, and it's 3' over here, and it's going to be 5' over here. So still DNA can only be replicated in this direction. It cannot go backwards just because of the shape of the nucleotides and how we remember that DNA is antiparallel. It can only be attached in a specific way, and it actually needs something. It needs like a shoulder to attach to before it can move on. And in the leading strand, the shoulder is another nucleotide because it's going in the right direction. However, there's no real shoulder to attach to, and that's what the RNA primer is used for. So the RNA primer makes like kind of like a foothold for our DNA chunks to line up and create a segment. Now remember, this segment right here, it's a little fragment of the daughter DNA, and that's called an Okazaki fragment. And it continues in here until it hits another RNA primer. But then what happens is this DNA ligase, this is another superstar, I'm going to put this over here, DNA ligase is actually what stitches all these little fragments together, so we get a nice beautiful strand just like the leading strand, and we don't have any of these RNA primers stuck in place. So let's write that out for step number 5. RNA primer is replaced by DNA nucleotides. And DNA ligase joins together Okazaki fragments. Now, I have a video. This is kind of hard. This was important so that you know who's who in this whole lagging strand conundrum here. But I have a video that is actually an animation of this, and that's going to show you kind of how this is done, and it's easier to understand this if you can actually physically see it happening. So really quick in the review on the lagging strand, since DNA can only be replicated from 5' to 3', the replication is moving away from the replication fork. And that gives us an issue because of the way that nucleotides chemically are. They need something to attach to in order to really exist on this strand here, on the parent strand. So to solve that problem, we have an RNA primer making this attachment point for DNA nucleotides to attach and to create a daughter strand, or a part of the daughter strand, going in the correct direction, 5' to 3'. And at the end, you have this kind of strand that has all these little fragments called Okazaki fragments, and they're stitched together by DNA ligase, and they make another beautiful daughter strand. So that's our leading strand versus lagging strand conundrum. Be sure to watch the animation video below and write down any questions you have. Thank you. Bye-bye.